Hello, everyone. My name is Malcolm Guzman. I'm a member of Group 3, and today we're going to talk about fluorescence resonance energy transfer. So what is fluorescence resonance energy transfer, or FRET? Well, let's say we have a protein, fluorescent protein, such as CFP or cyan fluorescence protein, which has an excitation energy of 440 nanometers and an emission wavelength of 490 nanometers. And then we place it really close to another protein, such as YFP or yellow fluorescence protein, which has an excitation wavelength of 490 and an emission wavelength of 527 nanometers. So you'll notice that the emission wavelength of CFP and the excitation energy of YFP overlap, which means that when we expose cyan to 440 nanometers light, then that will cause YFP to produce light in its own wavelength. And so that's how we can tell that these two proteins are really close together. And there we have the energy transfer in fluorescent resonance energy transfer. Now, how is this useful? Well, let's say we have two proteins that are marked with the cyan fluorescent protein and the yellow fluorescent protein, and they interact in really close proximity. Well, then when we expose this whole complex to 440 nanometers of light, then that will cause the yellow protein to also emit light. And this can be used to observe the interactions between different proteins, or it can be used to observe interactions of different parts of a single protein, which both have the fluorescent proteins attached, which can be really useful in sort of determining protein interactions and different processes in the cell. How is this applied? Well, as we talked about with the interaction of different parts of a single protein, we have what are called chameleon biosensors. Basically what these are proteins that change shape whenever they are exposed to high concentrations of calcium. And they have also the attached cyan and yellow fluorescent proteins. And so when the protein is exposed to calcium, it will change shape such that the yellow protein and the cyan protein come into close proximity and therefore emit light. So when we see the light, that means that there's a lot of calcium. It can also be used in really interesting ways, and not only in just identifying other protein interactions within the cell, but also in solar cells, where we can actually use the same process to extend the wavelength that a certain organic solar cell can absorb by taking advantage of those overlapping excitation and emission wavelengths, which I thought was pretty interesting and pretty useful outside of cell biology. Let me go back up here. So in this case, we have a graph from an article that I found called An Easy-to-Use FRET Protein Substrate to Detect Calpain Cleavage in Vitro and in Vivo. Now, calpain is a protease that is designed to cut its substrates in a way that is transformative and irreversible. Excessive calpain activity has been identified as playing a role in stuff like Alzheimer's or certain cancers, and so it's really important to analyze and be able to identify such calpain activity. Now, what this experiment was trying to find is to use FRET to identify this sort of activity. And in this chart, we can see basically what they did was take this PLFAAR sequence, which is really good at being cleaved by calpain, and they flanked it with cyan and yellow on either side. They then we're trying to find the optimal length for this sequence to be cleaved by calpain. And so they started adding serine, lysine, and alanine to it in different numbers of residues. And so we have 12, 14, 16, 18, 20, and 22. And then they measured the emission of cyan when it was exposed to a certain wavelength and the emission of yellow when it was exposed to a certain wavelength. And they observed that there was this inverse correlation when they added more and more bases, where yellow was emitting less light because it was absorbing less light from cyan, and that meant that these two proteins were moving farther and farther apart, which meant that the sequence was indeed getting longer without compromising its molecular integrity, which is what they were trying to do, because they were trying to find the right length, but it would be the best length for it to be cleaved. And they were making sure that adding serine, glycine, and alanine would be an effective way to do that. And this led them to observe that FRET is a really good way to identify calpain activity as opposed to other antibody-based calpain identifiers. So my question with this is, does the emission and absorption of light by fluorescent proteins affect the energy in cellular reactions? And if so, can this be harnessed in useful ways, such as when I mentioned the whole thing about solar cells? Like, is this changing activation energy of certain reactions, or is this exchange of energy interacting in a meaningful, interesting way that we can then harness to improve certain cellular processes or anything like that? So that's about it for me. That has been fluorescence resonance energy transfer. I thought it was really interesting, and I hope you guys did too. Thank you very much. Have a great day.